Hello students, welcome to EPG Pathshala. I am Dr. Rajiv Jain from Jivaji University Gwalior. Today we are going to discuss about the module Automatic Analyzers and Process Control under the paper Fundamentals of Analytical Chemistry. Here we shall discuss different types of automatic analyzers, how to interface them with different types of laboratory instruments and also instruments present in different types of industries. Analyzers were designed for rapid analysis of samples. Process analyzers depend to a large extent on the sampling system. For example, process gas chromatography is used widely for the analysis of petroleum and petrochemical products. Gas chromatograph is one of the important process analyzers which has been attached with different types of processes. Infrared analyzers for processes in different types of applications are quite different from the laboratory counterparts. Moisture analysis for gases is the electrolytic hygrometer. Automatic analyzers earlier were designed for repeated analysis of the samples so that number of samples can be done uh, in a very small time, so a small period. These instruments duplicate exactly as possible volumetric titration procedures used by practicing analysis. Repeated cycles of operation with a single reaction vessel is very time consuming. The need to analyze large number of samples in clinical laboratories has resulted in renewed interest in the development of automatic analyzers. However, these instruments generally have many things in common. Specific color forming reagents and analysis steps are carried out sequentially as the sample moves through the instrument. As, as you can see in figure 1, a number of samples are held in a rotating sample tray. Transfer of a fixed aliquot of one of the sample to a sample cup. The cup with the sample aliquot then moves to the next position where predetermined amount of the reagents are added, then stirring and if necessary filtration takes place. The sample cup now moves into an incubator to develop color characteristic of the sample component of interest. Sufficient time has been allowed for color development. Sample is transferred to a colorimeter and intensity of color is measured at a fixed wavelength. This provides a measure of the concentration of the sample component, and in the figure 1 all these things has been explained how automatic analysis can take place. The instrument is normally set up to analyze for the same component in a number of samples. However, there is a large degree of flexibility in the arrangement and different components can be determined by the use of different reagents. In some cases, it may be necessary to add extra components, for example, to provide for the further addition of further reagents after the filtration step. This is facilitated by constructing the instrument on a modular basis and at any time using only those modules required for their specific analysis. The methods used for various steps, such as reagent addition, stirring, and filtration, depend on the manufacturer of the instrument. In the auto analyzer, as shown in figure 2, an alternative approach is adopted. Instead of taking a fixed aliquot of each sample for the analysis, a constant flow of sample is used. The first application for this instrument was the determination of urea nitrogen in blood, and a diagram of the instrument being used for this purpose is shown in figure 2. There have been a number of changes made in the chemical system used for this particular analysis, mainly to improve quantitative results. In the first system developed, the urea was incubated at 55 degrees centigrade with a buffered urea solution, and the resultant ammonia was picked up by a water stream in the dialyzer. To this stream was added a solution of Nessler's reagent that produced a yellow color in proportion to the concentration of urea in the sample. In the last version, diacetyl monooxime hydrolyzers in acid solution to form diacetyl. The diacetyl reacts with the urea to produce a yellow diazine derivative, and the color is intensified by the addition of thiocyanate that forms a red triazine derivative as shown in the following figure. The reaction rate is speeded by heating the solution to 90 degrees centigrade. The light transmitted is then measured at 520 nanometer in a 15 millimeter flow cell. In figure 2, the diacetyl monoxide solution contains thiocyanate and the acid solution is 20% sulfuric acid containing ferric chloride and phosphoric acid. Chloride ions sensitize the reaction and phosphate ion increase the reproducibility of the reaction. The heart of the autoanalyzer is a peristaltic pump which has up to 28 separate flexible pumping tubes. The sample and reagents are forced through the tubes by means of transverse rollers that continuously squeeze the fluids ahead of them. Different flow rates can be obtained by using tubes of different internal diameter. Since the ratios of the volumetric rates of reagent and sample remain constant, there is no need for measuring out fixed aliquots, and automatic pipettes are therefore not required in this instrument. Sample and reagents are mixed by passing them through a series of horizontal glass coils. In place of filtration, a dialyzer is generally used. This consists of two matching grooves cut in a pair of transparent plastic plates. The plates are clamped together with the grooves separated by a cellophane membrane. The sample flows through the upper half and diffusible constituents pass through the cell phone semi-permeable membrane into the flowing stream of reagent in the lower half. The amount that diffuses through the membrane is proportional to the concentration but depends on the sample component. Next thing is the color development and measurement. Color development occurs in a heating bath which is set at about 95 degrees centigrade. The color forming reactions may be relatively slow and the stream containing the sample and reagent therefore flows through a glass helix approximately 33-meter long which is immersed in the bath. The time taken to traverse the helix is about 5 minutes. From here, the stream flows to the calorimeter and the concentration of the sample component is continuously displayed on a pen recorder. As an alternative to the calorimeter or a fluorometer is used for some clinical analysis and a flame photometer for the determination of sodium and potassium. Autoanalyzer is also used for the sequential analysis of a number of samples. These are placed in a rotary sample tray, and as the tray rotates, a metal probe dips into each sample cup and aspirates the sample for a fixed time interval. Between zero point 0.2 and zero point 0.6 ml of sample is normally used depending on the speed of the rotation of the sample tray. 20, 40 or 60 samples per hour may be taken. The probe then lifts out of the cup, aspirates air from one second and goes into a wash receptacle. After that, it lifts out of the wash solution, aspirates air again, and then moves into the next sample cup. Each sample is thus separated by a segment of air and by a wash solution. Air is also introduced into the fluid lines to segment the reagent streams. This segmentation of the fluid streams provides a wiping action that removes traces of residual material from the tube walls, thereby further preventing cross contamination between samples. However, the air bubbles must be removed before the fluid stream enters the column. Specific and non-specific analyzers. Process analyzers can be divided broadly into two classes, specific and non-specific. In non-specific analyzers, some bulk property of the process stream is measured, for example, viscosity or thermal conductivity. This is generally useful as an analytical method only for binary mixtures, but it is the simplest, cheapest, and more reliable method. An exception to the binary mixture limitation occurs where the contribution from one component in the mixture differs markedly from that of all other components. The position is particularly favorable when its contribution is considerably greater than that of the other components. Specific analyzers are capable of measuring one or more individual components in a sample mixture, and a number of these instruments will be discussed in this lecture. In principle, any laboratory instrument can be adapted for process use, but in practice, special instruments are required to meet plant requirements. The need for continuous analysis has also resulted in the development of a number of analyzers that are not normally found in the research laboratory. Because process analyzers must function unattended for extended periods of time, they must be more reliable and more stable than the corresponding laboratory instruments. Furthermore, chemical plants provide a relatively hostile environment for analytical instruments. On the one hand, the analyzer must be protected from corrosive materials within the plant. A block diagram of a typical process analyzer installation is shown in figure 3. It consists of a sample conditioning system usually located on the plant, the analyzer, which is usually located in an enclosed analyzer house and has associated with it any necessary gas supplies, reagents, and calibration samples. The output system, usually located in the plant control room, a programming unit which controls the operation of the analyzer, is also required for repetitive instruments such as the chromatograph. Sample handling system. Successful results from a process analyzer depends to a large extent on the sampling system. This must present to the analyzer a representative sample in an acceptable form with a minimum of delay and at a suitable flow rate. The location of the sample point must be chosen carefully to ensure that the sample is representative of the process stream. If the process stream is hot, the sample line will probably require heating to avoid loss of some of the sample components by condensation in the line. Heating is provided by steam jacketing the line or by steam tracing, that is, by running a stream line adjacent to the sample line. The function of the sample conditioning system is to treat the sample so that it is acceptable to the analyzer. For the simple case of gas under positive pressure, the sampling system may be no more than a filter and pressure reducer. In general, additional components may be necessary for heating or cooling the sample, drying it, removing entrained gas or liquid droplets, and for scrubbing our corrosive or other unwanted substances. At the same time, it is important to ensure that the components of interest are not removed by the sample handling system. When sample streams need extensive cleanup, the sample conditioning system may resemble a small chemical processing plant. The sample flow rate required depends on the particular analyzer, but it would typically be around zero point 0.1 to 11 per minute. This determines the size of sample conditioning system. To reduce the time required for the sample to travel from the sampling point to the analyzer, A much larger sample flow is normally taken from the process stream, and most of this is returned to a lower pressure point in the pressure via the sample bypass line. Delays in the sampling system are also minimized by making the sample lines as short and narrow as possible. As I told you in the beginning of the lecture, that process gas chromatograph is one of the most important process analyzers and also provides an excellent example of a complete analyzer system. Here, the important thing is that samples are automatically transferred one by one to the column by auto hydrodynamic syringe. Another feature of many process chromatographs is the use of multiple columns to enable complex samples to be analyzed. The principle of the automatic sample valve is shown in figure 4. In the version shown here, there are two discs. The upper disc contains inlet and outlet tubes and the sample loop or tube that determine the size of the sample, usually about 0.1 to 1 ml of gas. This disc is normally made of stainless steel and its bottom face is polished optically flat. The lower disc is of PTEF and seals against the polished face of the upper disc. As shown in the figure 4, it has three channels cut into its surface that link together the inlet, outlet, and sample tubes. In position A, the flowing sample fills the sample loop. In position B, the lower disc has been rotated at 60 degrees centigrade, thereby interchanging the tubing connections and introducing the sample tapped in the same loop into the carrier gas line. With liquids, very small volumes are involved, only a few microliters. For this reason, liquid samples are usually vaporized and a vapor sample is injected into the column by means of a heated gas sample valve. Columns and detector. The characteristics of chromatograph columns used for process instruments must remain stable for an extended period of time. Consequently, some column operating conditions that are acceptable in the laboratory are unsuitable for process chromatograph. The use of multiple column system in process chromatograph is quite common, particularly for the removal of unwanted high boiling components. This procedure, which employs a stripper column, is illustrated in figure 5. At the start of the analysis, the stripper and analysis column are in series as shown in figure 5a, and this configuration is retained until all the wanted components have been passed through the stripper and into the analysis column. The carrier's gas flows through the stripper column, is then reversed, figure B, and the heavier sample components are backflushed to vent. At the same time, the carrier gas flow through the analysis column is maintained constant by introducing a flow restrictor into the carrier gas line in which place of the stripper column. Column switching system involving up to 3 or 4 or more columns may be required for rapid analysis of samples containing a large number of components or for the separation of important trace impurities. As with laboratory instruments, the two detectors most commonly used in process gas chromatograph are the thermal conductivity detector and the flame ionization detector. The latter provides the sensitivity required to analyze for trace quantities of hydrocarbons and can also be used for carbon monoxide and carbon dioxide at ppm level by converting these compounds to methane over a nickel methanation catalyst before detection. The oven and detector assembly is either housed in a flame-proof case or is purged with a continuous flow of air to meet plant safety requirements. Programmer and readout system. Column switching and repetitive injection of the sample are controlled by a programmer. This sequencing or timing device also presets the signal attenuation for each peak, controls the operation of the recorder, and automatically re-zeros the baseline prior to each analysis. Various programming devices have been developed, but those most commonly used with process analyzers employ either manually set timings, cams, or a multi-channel rotating disc and photoelectric pickup head together with associated switches. The relationship between the chromatogram and the programmer timing functions is shown in figure 6. The normal chromatogram is unsuitable for repetitive display of data, and it is usually converted into a bar graph form as shown in the figure 6. This is done by keeping the recorder paper stationary during the emergence of each peak and stepping it in on a fixed distance between each peak. Alternatively, a multi-point recorder can be used with a different color or shade point for each component in the sample. Applications of process chromatograph. Most of the applications of process gas chromatograph are generally in petroleum and petrochemical industries for the formation of different types of products which are analyzed at every time of the product formation. Even with a single, it has been noted that in a single petrochemical industry, at a point, more than over 60 chromatographs has been used as an interface. Other applications include the monitoring of annealing furnace and blast furnace atmospheres, air annealing furnaces and blast furnace atmosphere, air separation processes, ammonia synthesis, helium in natural gas, and sulfur recovery. Process gas chromatographs can also be used in the research laboratory to monitor research reactors. Another important analyzer is oxygen analyzers. The particular method employed depends to a large extent on what and whether the measurement is for oxygen in the gas phase or for dissolved oxygen, and on the detection limit required. And it is based upon the measurement of electrical conductivity, as you can see from the following equation. One of the most sensitive methods for dissolved oxygen is based on the reaction and the subsequent determination of the ion concentrations by measurement of the electrical conductivity of the solution. On the other hand, the toxicity of thallium creates a potential problem. This instrument is used mainly for the determination of oxygen in boiler feed water. Another types of analyzers include electrochemical cells. Several electrochemical cell systems are used in commercial oxygen analyzers. An example is shown in figure 7, which is basically a holographic oxygen electrode. The silver anode and gold cathode are separated from the sample by a thin PTFE membrane. This is permeable to oxygen. With a potential of 0.8 volt applied between the anode and cathode, the following reaction takes place in the presence of oxygen. At cathode, oxygen, by taking 4 electrons, is converted into OH ions. At anode, silver reacts by chloride ions and forms silver chloride, and 4 electrons are released. The resulting current flow is proportional to the partial pressure of oxygen in the sample. Because the rate of diffusion on oxygen through the membrane varies by about 3% per degree, a thermistor is built in automatically adjust the output signal to compensate for temperature changes. The speed of response of the sensor is also determined by the oxygen diffusion rate. Typically, 90% of the final response occurs within 10 seconds. The device can be used for both dissolved oxygen and gaseous oxygen, but the sample must be free from other gases that are reduced at approximately zero point 0.8 volts. These include sulfur dioxide, chlorine, bromine, iodine, and oxides of nitrogen. Since the sensor consumes a small amount of oxygen and the diffusion of oxygen through liquid is relatively slow, a liquid sample must flow continually past the sensor for valid results to be obtained. With a static liquid sample, the oxygen supply would be gradually depleted. The device will cover the ranges 0 to 2.5 parts per million up to 0.100 ppm of dissolved oxygen or 0 to 500 up to 0 to 1 volume by volume of gaseous oxygen. Negative filtering infrared analyzer. In a negative filtering infrared analyzer, as shown in figure 9, a sensitizing cell that completely absorbs radiation corresponding to the component of interest is placed in front of one half of the detector. When the component is present in the sample, the intensity of the characteristic radiation passing through the sample cell is reduced. The effects, this affects si- only the side of the detector preceded by the chamber containing the non-absorbing gas, and the difference signal between the two halves of the detector is reduced. Because infrared absorption bands are not unique for each component, there may be unwanted absorption by other components in the sample and interferes with the measurement. This is the figure which explains the things in figure 9. Infrared negative analyzer has been shown. What are the applications of infrared analyzers? One of the major applications for the infrared gas analyzer is the determination of carbon monoxide and carbon dioxide. It is very important. In atmosphere, when we want to determine carbon monoxide, this type of infrared gas analyzers are used. Continuous analysis for these responses, for these components are required for the control of ammonia, synthetic plants, boilers, blast furnaces, and metal treatment plants. Other important applications of the infrared analyzers are in air pollution studies and vehicle exhaust emission control, as I have told you, in the determination of carbon monoxide and carbon dioxide gases. Another type of analyzer is moisture analysis. There are approximately 20 different methods for the automatic determination of moisture in solid, liquids, and gases. These include automatic titration procedures for liquids and infrared absorption for gases. Electrical capacitance measurement can be used to continuously monitor the water content of sheet material and microwave absorption by granular solid. We shall concern ourselves mainly with the measurement of moisture in gases. The commonest possible moisture analysis for gases is the electrolytic hygrometer. In this instrument, there is an electrolytic cell which consists of two closely spaced platinum or rhodium wires approximately zero point 0.3 mm apart, wood on a cylindrical former and coated with a film of phosphorus pentoxide. This film absorbs water from the gas sample, and the absorbed water is continuously electrolyzed by a DC voltage applied between the two wires. At a constant sample flow rate and with all the water removed from the sample stream, the electrolysis current is a direct measure of the water content of the sample gas. The instrument can therefore be calibrated directly by application of Faraday's law. In spite of the fact that response of the electrolytic hygrometer depends on the electrolysis of absorbed moisture, approximately 90% of the final response is reached in 1 minute with a sample flow rate of 100 ml per minute, provided that the change in moisture content is not too large. Rhodium is preferred as the electrode material because it minimizes the errors that occur due to the recombination of the electrocatalytically generated oxygen and hydrogen in streams containing high concentrations of hydrogen. It also increases the operating life of the cell at high moisture concentrations. The instrument is suitable for measuring moisture over the range 0 to 10 to 0 to 1000 ppm in inert gases, hydrocarbon gases, and even in chlorine. However, it cannot be used with basic gases such as ammonia or with certain organic materials such as alcohols and acetone. The phosphorus pentoxide film also catalyzes polymerization of some unsaturated organic, particularly conjugated dienes such as butadiene. To overcome this problem, a gas chromatograph can be used in conjunction with the electrolytic hygrometer to carry out a preliminary separation of the unwanted sample components. In measuring low concentrations of moisture in gas samples, considerable care is necessary to exclude atmospheric moisture. Rubber and many plastics are permeable to moisture and therefore cannot be used. Small leaks in the system cannot be tolerated because atmospheric moisture will back diffuse in the system even against a high sample pressure. This occurs because the moisture diffusion rate is determined by the difference between the partial pressures of water vapor in the sample and in the surrounding atmosphere and not by the difference in the total pressure. Similar considerations also apply to oxygen analyzers. Moisture is absorbed readily by many materials and desorption can give rise to errors and that persist for extended period of time. Calibration of moisture analyzers other than the electrolytic hygrometer presents a problem, but samples containing low concentrations of water can be produced by the catalytic conversion of hydrogen and oxygen to water. A moisture-free gas containing the appropriate hydrogen plus X is oxygen, for example, 100 ppm hydrogen and 1 to 2% oxygen in nitrogen will remain stable in a gas cylinder for an extended period and can be used as required. Students, we have learned in this lecture what are automatic analyzers, what are process control analyzers, what are their applications, how they can be interfaced, particularly in industries where there is corrosive environment. So for environment is different in industries, corrosive environment, and in laboratories environment is different. So for putting the instrument in level in an industry so that it can be directly interfaced with the process taking place in the different types of industrial reactors and then transferring the compounds, the synthetic products at every stage to the process analyzers is very important how it is transferred there. Then after transfer at every stage results are obtained. For example, different types of analyzers include typical process analyzers, oxygen analyzers, calorimetric analyzers, moisture analyzers, infrared analyzers, elemental analyzers, gas chromatograph, mass spectrophotometers, all can be interfaced with the different types of reactors processes taking place. Different type of methods were used in process control, all you have seen in this module. Thank you.